Hello and welcome, dear students. Today our topic is tRNA and its role. The main objectives of this module are to know about the structure of tRNA, to know about the biogenesis and tRNA genes, to know about the types of tRNA and the post-translational modifications of tRNA, and finally to know about the functions of tRNA. Dear students, let's first discuss what is tRNA. Transfer ribonucleic acid or tRNA is a type of RNA molecule that helps decode a messenger RNA, that's mRNA sequence, into a protein. The term tRNA was coined by Allen and Shewit in 1960 and used to describe a group of low molecular weight RNA molecules that play a vital role in protein synthesis or translation. The purpose of tRNA or transfer RNA is to bring amino acids to the site of ribosome for protein production. To make sure that the amino acids are added to the protein in a specific order, the tRNA reads the codons from the messenger RNA or mRNA. Each tRNA carries a specific amino acid based on its anticodon. The tRNA molecule serves as an adapter between the genetic instructions written in nucleic acid sequence in the gene and the protein products encoded in the genes. In other words, we can say that tRNA is a small molecule or small RNA molecule that plays a key role in protein synthesis. It serves as a link or adapter between the messenger RNA or mRNA molecule and the growing chain of amino acids that make up a protein. Each time an amino acid is added to the chain or growing polypeptide chain, a specific tRNA pairs with its complementary sequence on the mRNA molecule, ensuring that the appropriate amino acid is inserted into the protein being synthesized. The existence of this type of molecule had been predicted by Francis Crick in 1955, who was unable to envisage DNA or RNA acting as direct template for amino acids. Crick suggested that adapter molecules like tRNA were necessary to translate the information carried by nucleic acids into a protein. In its simplest form, the hypothesis required that there was one adapter molecule for each amino acid, each molecule having a specific hydrogen bonding surface that would enable it to bind specifically to a nucleic acid template, and one enzyme for each amino acid that would be responsible for the specific attachment of the amino acid to its adapter molecule. Transfer RNAs, then termed soluble RNAs or sRNAs, because they occur in the soluble cytoplasmic fractions of cells, were assigned the role of adapter molecules by Hoagland et al. In 1958. Later on, his investigations were stimulated by a desire to understand the mechanism of translation in structural terms. When in the early 1960s, techniques for determining the nucleotide sequence of RNA molecules were developed, the tRNA, that is the smallest species of RNA present in the cell, attracted further attention as relatively simple, naturally occurring nucleic acids, which it was hoped would help in the elucidation of the general principles governing nucleic acid protein interactions. In the past decade, the sequence of nearly 100 tRNAs have been determined and their structures have been further investigated by chemical and physical techniques of molecular biology. Study of biological role of tRNA has not only given a fuller understanding of the mechanism of protein synthesis, but has also shown that the role of tRNA in the cell is not confined to that of an adapter molecule only. However, attempts to relate the structure of tRNA even to its principal known function as an adapter have met with mixed success. At present, although we have some hints of the structural features of the tRNA required in some of its functions, much remains unclear. One of the major difficulties in relating tRNA structure to its function was that until recently, the three-dimensional structure of tRNA was not known in detail. Physical and chemical studies of tRNA in solution were limited to providing structural data which was either at a low resolution, giving only an indication of general shape, or resolved to the nucleotide level but restricted to selected regions of the molecule. In 1974, the three-dimensional structure of yeast tRNA was derived from X-ray diffraction analysis of two different crystalline forms. This three-dimensional model represents a static form of the tRNA molecule which functions dynamically in the cell, but it provides a structure in sufficient detail to allow us to ask specific questions about tRNA function in structural terms. Dear students, let's now discuss the structure and functions of tRNA. Since the first nucleotide sequence of a tRNA was determined by Holle et al. In 1965, nearly 100 different tRNAs have been sequenced. This accumulating data quickly provided strong circumstantial evidence for a common secondary structure, since all the sequences determined could be accommodated within a cloverleaf structure. In addition, sequence information has proved invaluable in establishing other common structural features of tRNA, thus helping our understanding of some structure-function relationships, as well as suggesting features of the three-dimensional structure of yeast tRNA, which are most probably common to all tRNAs. Transfer RNA tRNA range in length between 70 and 100 nucleotides. tRNA are acylated with the cognate amino acid by their cognate aminoacyl tRNA synthetase, and the resulting aminoacyl tRNAs are substrates for ribosomal protein synthesis. tRNAs were determined early on to have a highly conserved cloverleaf secondary structure and an L-shaped tertiary structure. The cloverleaf secondary structure is formed from Watson-Crick base pairs which create helical stems, typically ending in unpaired bases to form loops. These arms or stem and loop include the acceptor arm, D arm, anticodon arm, T pseudouridine C arm, and a variable arm. Of these features, the acceptor arm, anticodon arm, and T arm are highly conserved in size, while the D arm and variable arm can differ. The anticodon loop, which pairs with mRNA, determines which amino acid is attached to the acceptor stem. The anticodon loop is recognized by aminoacyl tRNA synthetase enzyme that chemically links a tRNA to an amino acid through a high energy bond. Aminoacyl tRNA synthetase reads the anticodon and also recognizes the D arm located downstream from the 5' end of the tRNA molecule. The D arm is made of a double-stranded stem region formed by internal base pairing as well as a loop structure of unpaired nucleotides. The D arm is a highly variable region and plays an important role in stabilizing the RNA tertiary structure and also influences the kinetics and accuracy of translation at the ribosome. The other structure that influences the role of tRNA in translation is the T-arm. Similar to D-arm, it contains a stretch of nucleotides that base pairs with each other and a loop that is single-stranded. The paired region is called the stem and mostly contains 5 base pairs. The loop contains modified bases and is also called the T-pseudouridine C-arm to specify the presence thymidine, pseudouridine, and cytidine residues, that is the modified bases. tRNA molecules are unusual in containing a high number of modified bases as well as containing thymidine, usually seen only in DNA. The T-arm is involved in the interaction of tRNA with the ribosome. Finally, a variable arm containing less than 20 nucleotides is situated between the anticodon loop and the T-arm. It plays a role in the amino-acyl tRNA synthase recognition of tRNA, but could be absent in some species. The stems of the acceptor arm and T-arm are found to have 7 and 5 base pairs respectively to give the canonical 7/5 configuration. Occasionally, a base pair mismatch is found in the acceptor stem, which does not disrupt the conformation of the helix. Furthermore, the acceptor arm has 4 3' terminal residues which are not base paired: the discriminator base and the CCA tail. The 3' terminal adenosine is in the form of a slightly activated ester to bind to the amino acid. The 3' strand of the acceptor stem is directly connected to the T stem, while the 5' strand is connected to the D stem by 2 unpaired bases. The length of the D stem varies amongst tRNAs. Most of them have between 3 and 5 base pair leading up to the D loop. The anticodon stem is also highly conserved in length, being found to consistently have 5 base pair before ending with the anticodon loop. The anticodon loop has 7 nucleotides with the 3 residues in the center of the anticodon loop. Participating in mainly Watson-Crick but also sometimes non-Watson-Crick or vowel base interactions with the codon of the mRNA. Finally, the variable loop is the least conserved amongst all tRNAs. Dear students, let's discuss the anticodon. An anticodon is a unit of 3 nucleotides corresponding to the 3 bases of an mRNA codon. Each tRNA has a distinct anticodon triplet sequence that can form 3 complementary base pairs to 1 or more codons for an amino acid. Some anticodons pair with more than 1 codon due to vowel base pairing. Frequently, the first nucleotide of the anticodon is one not found on mRNA, inosine, which can hydrogen bond to more than 1 base in the corresponding codon position. In genetic code, it is common for a single amino acid to be specified by all 4 third position possibilities, or at least by both pyrimidines and purines. For example, the amino acid glycine is coded for by the codon sequence GGU, GGC, GGA, and GGG. Other modified nucleotides may also appear at the first anticodon position, sometimes known as vowel position, resulting in subtle change to the genetic code, as for example in mitochondria. Per cell, 61 tRNA types are required to provide one-to-one correspondence between tRNA molecules and codons that specify amino acids, as there are 61 sense codons of the standard genetic code. However, many cells have under 61 types of tRNAs because the vowel base is capable of binding to several, though not necessarily all, of the codons that specify a particular amino acid in the genetic code. At least 31 tRNAs are required to translate unambiguously all 61 sense codons which code for amino acids. Biogenesis of tRNA. In eukaryotic cells, tRNA are transcribed by RNA polymerase III as pre-tRNAs in the nucleus. RNA polymerase III recognizes two highly conserved downstream promoter sequences, the 5' intragenic control region, that is 5' ICR, decontrol region or A box, and the 3' ICR, that is T control region or B box, inside the tRNA genes. The first promoter begins at the +8 of mature tRNAs, and the second promoter is located 3060 nucleotides downstream of the first promoter. The transcription terminates after a stretch of 4 or more thymidines. Pre-tRNA undergoes extensive modifications inside the nucleus. Some pre-tRNAs contain introns that are spliced or cut to form the functional tRNA molecule in bacteria. These self-splice, whereas in eukaryotes and archaea, they are removed by tRNA splicing endonucleases. Eukaryotic pre-tRNAs contains bulge helix bulge or BHB structure motif that is important for recognition and precise splicing of tRNA introns by endonucleases. This motif position and structure are evolutionarily conserved. However, some organisms such as unicellular algae have a non-canonical position of BHB motif as well as 5' and 3' ends of the spliced intron sequence. The 5' sequence is removed by RNase polymerase, whereas the 3' end is removed by the tRNase Z enzyme. A notable exception is in the archaean, nanoarchaean quitons, which does not possess an RNase P enzyme and has a promoter placed such that transcription starts at the 5' end of the mature tRNA. The non-templated 3' CCA tail is added by a nucleotide transferase before tRNA are exported into the cytoplasm by LOS1 export T. tRNAs are amino acylated. The order of the processing events is not conserved. For example, in yeast, the splicing is not carried out in the nucleus but at the cytoplasmic site of mitochondrial membranes. In March 2021, researchers reported evidence suggesting that a preliminary form of transfer RNA could have been a replicator molecule in the very early development of life or abiogenesis. Dear students, let's now discuss the tRNA genes. Organisms vary in the number of tRNA genes in their genome. For example, the nematode worm C. elegans, a commonly used model organism in genetic studies, has 29,647 genes in its nuclear genome, of which 620 code for tRNA. The budding yeast Saccharomyces cerevisiae has 275 tRNA genes in its genome. In the human genome, which according to January 2013 estimates has about 20,848 protein coding genes in total, there are 497 nuclear genes encoding cytoplasmic tRNA molecules and 324 tRNA-derived pseudogenes, tRNA genes thought to be no longer functional, although pseudo-tRNAs have been shown to be involved in antibiotic resistance in bacteria. As with all eukaryotes, there are 22 mitochondrial tRNA genes in humans. Mutations in some of these genes have been associated with severe diseases like the MELAS syndrome. Regions in nuclear chromosomes very similar in sequence to mitochondrial tRNA genes have also been identified. The phenomena of multiple nuclear copies of mitochondrial tRNA, that is tRNA look-alikes, has been observed in many higher organisms from humans to the opossum, suggesting the possibility that the look-alikes are functional. Cytoplasmic tRNA genes can be grouped into 49 families according to their anticodon features. These genes are found on all chromosomes except the 22nd and Y chromosome. High clustering on 6p is observed, that is 140 tRNA genes, as well as on chromosome 1. Dear students, let's now discuss the types of tRNA. A tRNA can be classified based on the amino acid it carries, giving rise to 20 different tRNAs. Alternatively, they can also be grouped based on their anticodon. There are 64 possible codons arising from a combination of 4 nucleotides. Of these, 3 are stop codons that signal the end of translation. This gives rise to a situation where one amino acid is represented by multiple codons and the amino acyl tRNA synthetase enzyme as well as the tRNAs have to accommodate this redundancy. However, very few species have exactly 61 tRNAs, which gives rise to the question of how every codon is recognized by a specific tRNA molecule. In many species, the number far exceeds 61, and different tRNAs carrying the same anticodon could display varying efficiency in translation, adding a layer of regulation to the process of protein synthesis. tRNAs interact with codons on the mRNA through their anticodon loop. Base pairing between the codon and anticodon ensures specificity during translation. However, the first base of the anticodon that pairs with the vowel or third position in a codon is often modified to allow the tRNA to hydrogen bond with 3 instead of 1 base. Thus, a single tRNA has the option of recognizing and base pairing with 3 codons which code for the same amino acid. There are 20 amino acyl tRNAs, one for each amino acid. This group of enzymes can recognize all the anticodons representing a particular amino acid and therefore acts as the second arm of the machinery that handles genetic code redundancy. Finally, these molecules can also be classified into 3 categories: those carrying canonical amino acids attached to the correct tRNA, and those that are incorrectly attached, and those carrying modified amino acids such as selenocysteine for non-canonical elongation. Dear students, let's now discuss the post-transcriptional modification of tRNA. There are nearly 500 genes coding for tRNAs in the human genome and 300 gene fragments associated with these RNA. These genes are transcribed by RNA polymerase III, and the transcript undergoes extensive modification, especially in eukaryotes. Introns are spliced or cut. The intron-exon boundary is acted on by endonuclease. The 5' and 3' ends of the RNA are processed, and enzymes add the terminal CCA residues to the 3' end of the tRNA molecule. The CCA site residues could become amino-acylated in the nucleus itself, and this charged tRNA could then be exported from the nucleus. Additionally, many bases on the tRNA are also modified, especially by methylation, that is addition of a methyl group, and deamidation, that is removal of an amide group. Particularly the first base of the anticodon that pairs with the vowel position on the codon is modified to allow unusual types of base pairing. Adenine can be modified to form inosine, which expands the pairing possibility to include uracil, cytosine, and adenine. Pseudouridine, on the other hand, is another common modified base derived from uridine residue through enzyme-mediated isomerization reaction. It is said to play a role in the structural integrity of the tRNA molecule, being involved in stiffening the nearby sugar phosphate backbone and also influencing base stacking of proximal regions. Lysine is an unusual base formed when a lysine amino acid is attached to the cytidine residue. Lysine pairs specifically with adenosine, a property that is used by the isoleucine tRNA to ensure translation specificity. Amino-acyl tRNA synthetase attach the appropriate amino acid to tRNA molecules based on their anticodon. These enzymes contain binding sites for the amino acid tRNA as well as ATP and hydrolyze ATP to AMP, that is adenosine monophosphate, and attach the amino acid to the ribose sugar of the last nucleotide on tRNA. The tRNA is now considered charged and can participate in the protein synthesizing reactions on the ribosome. This reaction often occurs in the cytoplasm, though it has also been observed in the nucleus. The ribosome contains three important regions: the P, that is peptidyl site containing the growing polypeptide chain, the A site or the acceptor site that receives a number of charged tRNAs, and the E or exit site through which a deacylated tRNA leaves the ribosome. These sites span both the subunits of the ribosome and are denoted as PP site or AA site, with the first letter referring to the site on the smaller subunit. For instance, the PP site binds to tRNA anchoring a polypeptide chain, while the AA site anchors an incoming charged tRNA molecule with an amino acid. The peptidyl tRNA on the PP site transfers the growing polypeptide chain to the tRNA on the AA site and undergoes deacylation. To continue the process of translation, the ribosome moves ahead by one codon at a time, making the tRNA on the PP site shift to transient PE configuration and then to the EE site before leaving the ribosome. Similarly, the tRNA on the AA site adopts a temporary AP binding conformation before setting at the PP site, ready for the next amino acid to continue translation. Dear students, let's now summarize the entire lecture. At the end, we are able to conclude that transfer ribonucleic acid or tRNA is a type of RNA molecule that helps decode a messenger RNA or mRNA sequence into a protein. tRNAs function at specific sites in the ribosome during translation process, which is a process that synthesizes a protein from an mRNA molecule. Proteins are built from smaller units called amino acids, which are specified by 3 nucleotide mRNA sequence called codons. Each codon represents a particular amino acid, and each codon is recognized by a specific tRNA molecule. The tRNA molecule has a distinctive folded structure with 3 hairpin loops that form the shape of a 3-leafed clover. One of these hairpin loops contains a sequence called the anticodon, which can recognize and decode an mRNA codon. Each tRNA has its corresponding amino acid attached to its end. When a tRNA recognizes and binds to its corresponding codon in the ribosome, the tRNA transfers the appropriate amino acid to the end of the growing amino acid chain. Then the tRNA and ribosome continue to decode the mRNA molecule until the entire sequence is translated into a protein. Beyond their canonical role during protein biosynthesis, tRNAs also perform additional functions in both prokaryotes and eukaryotes, for example, in regulating gene expression. Amino-acylated tRNAs have also been implicated as substrates for non-ribosomal peptide bond formation, post-translational protein labeling, modification of phospholipids in the cell membrane, and antibiotic biosynthesis. Most recently, tRNA fragments or TRFs have also been recognized to play regulatory roles due to their universally conserved L-shaped 3-dimensional conformation, which is stabilized by extensive secondary and tertiary structure contacts and modifications. tRNA molecules are among the most stable RNAs in a cell and are considerably more robust than mRNAs. For a long time, tRNA fragments were considered as non-functional degradation intermediates, but have now been recognized to be major RNA species in human cells for which regulatory roles are beginning to be discovered. It was also recently shown that tRNAs can act as an effective scavenger of cytochrome C, consistent with a role in regulating apoptosis. Dear students, it was all about today's lecture regarding tRNA and its role. Hope you have understood it well. See you next time with a new topic. Till then, take care. Goodbye.